So now I would like to introduce our webinar speaker, Dr. Jill Rowan, who joined PEPperPRINT in April 2016 as technical sales and marketing representative on the West Coast. For over 10 years prior, Jill has worked for research institutes, small startup companies, and large biotech and medical device corporations as an independent researcher, product, and marketing manager. Jill has managed small product portfolios, but also large ones comprising up to 5,000 products sold nationally and internationally. Prior to her time at the Scripps Research Institute studying B-cell responses in HIV and HCV vaccine design, she received her PhD in immunology at Tufts Medical School in Boston. She is an author of numerous peer-reviewed journal articles and received scholarships and grants fully supporting her educational and research career. So the title of the presentation today will be Antibody Validation and Cross-Reactivity Analysis by High-Density Peptide Microarrays. So again, if you want to ask any questions, please use the question pane at the bottom, and we will also share the slides and post-webinar file. So Jill, the presentation is yours. You can just take it away.
Okay, thank you very much, Karsten, for the introduction, and thank you all for joining. Again, sorry about the delays. Let's just get started. So for the agenda, I'm just going to start with a very brief overview of the company and describe our technology, and then dive right into the focus of the webinar, discussing this reproducibility crisis, which is due in part to commercially available antibodies. And then go into PEPperPRINT solution and describe our antibody validation cross-reactivity toolbox, and we'll wrap it up with a short Q&A. So PEPperPRINT is a privately held independent company headquartered in Heidelberg, Germany. It's located southwest of the country, about an hour's train ride from Frankfurt. The laser printed peptide microarray technology that it's based on was initially published in Science in 2007. We incorporated and had our first product launch in 2011, and now just 5 years later we have satellite offices in Cambridge, Mass., San Diego, California, Brazil, and Japan. We offer over 30 standard microarrays, and a majority of our business comes from customized microarrays in which we design and develop arrays based on particular researchers' needs. We are growing, and we offer solubilized peptide and peptide pools, which are clinical and research grade, and in co-development of a number of ELISA test kits that will eventually be used in a diagnostic setting. So the uniqueness of PEPperPRINT's technology is that it allows direct synthesis of peptides onto glass slides. So this allows the benefits of high flexibility in terms of its peptide content, high peptide density per array, dramatically reduces material consumption, and has fast production times. So briefly how it works is we have this large peptide microarray printer. It's around 4 to 5 times the size of a normal office laser printer. But instead of having 4 color cartridges, our printer has 20 amino acid cartridges in which you have the amino acid embedded in a toner in each of the cartridge. So how it works is we have glass slides that are placed into the printer, and the amino acids are directly printed onto the slides, melted and coupled so it sticks to the slides, wash, deprotect, and then this process is repeated over and over till eventually you have completed your peptide of interest. So this process of direct synthesis of peptide on a chip allows a competitive advantage that saves material time and cost to other technologies which require pre-synthesis of peptides and then spotting of those peptides on the chip. So this extra step increases the amount of time it takes to make the chip. It increases the cost since you're paying per peptide and not per chip. And probably most importantly, it increases the amount of peptides per chip. So essentially you get a lower resolution epitope mapping. So in conclusion, PEPperPRINT outperforms the competitor technology. So now onto the focus of the webinar. So many researchers think that antibodies are a major driver of what has been deemed the so-called reproducibility crisis, in which experiments can't be reproduced, so the results stemming from those experiments are unfounded. So in May 2015, Nature published this article kind of highlighting this epidemic and specific examples out in the field of when this really occurred. So take Dr. Rim. He's a pathologist at Yale. He developed an antibody-based test to detect a skin cancer, and he secured $2 million in funding to move the product to the clinic. But when he bought a new batch of antibodies, the team couldn't reproduce the results. Dr. Prasa from Mount Sinai, he spent years chasing this potential pancreatic cancer marker, and they bought a detection kit from a commercial vendor. They ended up wasting 2 years, $500,000 and thousands of patient samples until they realized that the antibody in the kit recognized a different cancer protein than they thought it was. So this really prompted the Human Protein Atlas Consortium from Sweden to develop a pretty robust study looking at antibodies available from commercial vendors, and they showed that less than 50% of these antibodies can be effectively used in immunohistochemical tests. So some of these researchers were really, the whole point of doing this article is really to spread the word and to make people understand that when you're buying an antibody from a company, that what is on the actual vial, the label, and what's actually inside the vial might be two different things. So then they wanted to go on and kind of figure out specifically what exactly were the problems or most common problems with these antibodies. And they found as cross-reactivity, variability, and wrong application. So with cross-reactivity, an antibody is supposed to recognize only a specific target protein, but a lot of times it's going to end up reacting with other proteins in the sample. Variability, different antibodies coming from different animals that are producing the antibodies behave and fold differently. So there's usually a lot of variability in the antibodies, and as you can imagine in a commercial setting, a lot of times it's so high throughput, they're constantly using different animal batches and so on and so forth. Then the wrong application. This is probably maybe you're most familiar with when you're ordering an antibody. There's a column that shows what assays that they are applicable to, whether it's flow cytometry, immunohistochemistry. Sometimes what they're putting is they don't have the actual data to support it. So because of this, in the research community, the pressure is really on to start to characterize currently available antibodies prior to doing your experiments. And Dr. Rama, who I mentioned in the previous slide, a lot of what he was saying, especially in the research, coming from a research background as well, it kind of resonated that the thought did cross his mind to take the time and money and resources to validate the antibodies, but he was under a lot of time, he was under a tight deadline, and he didn't have too much money to be spending, so he simply didn't do it. And he really wanted to emphasize to the research community that it's not worth compromising the integrity of your experiments and to validate your antibodies. So that's the bad news. So the good news is that, PEPperPRINT offers a solution, and what we've kind of wrapped up and put together in what we call a toolbox, an antibody validation toolbox. So for the remainder of the talk, I'm just going to be discussing this. So it consists of 3 tools. First is our PEPperMap Epitope Mapping, which includes linear and conformational mapping services. The PEPperMap Epitope Substitution Scans. So this allows identification of essential and variable amino acids. And then finally, our PEPperMap Antibody Cross-Reactivity Analysis, which is made possible through our development of our Human Epitome Microarray. So first, for our epitope mapping, I'm just going to walk us through a schematic of how this actually works. So you start with your amino acid sequence of interest of the antigen to which the antibodies bind. And then the sequence is then translated into overlapping peptides. We normally do 15 more peptides with a maximum overlap. So it's 14 amino acid overlap. As mentioned, we directly print those peptides onto a glass slide. Then we add a target antibodies that is specific for the epitope. And then a secondary antibody with a fluorochrome. And then you can put the array into a scanner. And here we use duplicates, so for each hit you see 2 spots. And then we have specialized peptide analyzer that will immediately tell you what hits, which calculates to the epitope or your consensus motif of the antibody. So for example, here in the top, this is an example of an array of a monoclonal sample. That's identifying just a single epitope, and then on the bottom a polyclonal sample that's identifying multiple epitopes. And then this green and red ribbon is controls, HA and flag controls. So let's look at this epitope mapping in the context of comparing to a standard microarray. So we performed 2 peptide arrays covering an antigen CMPA that was translated into 15 amino acids with the maximum overlap of 14 amino acids, which is PEPperPRINT, and then a standard that has a reduced overlap of 11 amino acids. And as you can see, so both of these chips were then challenged with a serum of a sclerosis patient that would react to CMPA. And what you can see with the PEPperPRINT, you have 2 clear separation of 2 adjacent epitopes, and you can easily determine the epitope length. Whereas the standard that has lower resolution and less overlap, you cannot clearly distinguish the 2 epitopes and only able to identify PTP as a consensus motif. So our technology is very flexible based on your needs. We can use monoclonal, polyclonal antibodies, synthetic or purified antibodies, plasma or serum samples, and any species or isotype imaginable. So just briefly about our chip quality and reproducibility. This is something we take very seriously. We have an ISO 9001 certified laboratory. We have standard operating procedures for each test assay with a standard serum. This is just showing an example of batch-to-batch values. As you can see, as a correlation coefficient of above 0.9, which is our, it's very typical, and it's our requirements to pass QC. Okay, now on to conformational epitope mapping. So many epitopes rely on protein folding and form conformational epitopes. For example, as shown here, this is a receptor CD20, which is expressed on B cells, specifically on B tumor cells. And it has 4 transmembrane domains and 2 extracellular, 1 small loop up here and another large loop up here. It's about 40 amino acids, and it's forms a conformational by a disulfide bridge. So, clearly anything against this conformational motif here would not be able to be identified by linear peptide arrays. So a common technique to circumvent that is to take the linear peptides and cyclize it, or kind of bend the peptides. And we do that. I mean, we optimize the assay in which we perform the linear epitope array as previously described, but then we add one more step where we cyclize the peptide onto the array. So if you're wondering, gee, does that really work? The answer is yes. We have had this on the market for about a year now. We have a number of customers that have used this assay successfully, and we also have a proof of concept case study that I'll just kind of briefly go over. So you might be familiar with rituximab. It's a common therapy for use of B-cell tumors, and it interacts with CD20. So in 2007, JPC published a co-crystal structure of rituximab and CD20, and they showed that it bound, rituximab bound to CD20 in this somewhere in this 15 amino acid loop. So we wanted to employ this conformational epitope mapping to get a more in-depth view on, and determine what the actual epitope was. So we developed an array with the 15 amino acid loop that CD20 and rituximab is known to bind, and we did the maximum peptide overlap, and then we challenged the array with rituximab. And as you can see here, with the linear, which was kind of a control, we did not find any response. But then when we cyclized at 7, 10, and 13 amino acids, we were able to identify an 8 amino acid epitope. So here at the 7 amino acid, it's a lower signal probably due to the actual length being 8 amino acid, and the increase in signal from 10 to 13 is probably due to the being more similar to the actual 15 amino acid loop. Okay, so now onto tool number 2. This is our PEPperMap epitope substitution scan. I thought I would just use the same case study of rituximab CD20 to illustrate how it works. So we wanted to get a closer view of the essential and conserved amino acids in the epitope of rituximab and CD20. So we did a full substitution scan of part of the extracellular domain of CD20. And what that means is for each of the 20 or so amino acids, we would substitute with with all the 20 amino acids. So it's it's more high throughput and robust than a typical alanine scan, which you're just substituting each amino acid for an alanine. So this gives 20 times more data points and gets much more clarity on what you are the essential and conserved amino acids. So here, as part of the extracellular domain, here is what we identified as the epitope. And as you can see, the epitope does not tolerate a lot of amino acid exchanges, and in particular, the MPS is absolutely essential, in which there is no toleration of any of the amino acid exchanges. So here you have it's one chip, one assay, very simple, and you clearly get a lot of information about the epitope and conserved and essential amino acids. Finally, on to tool number 3. It's our antibody cross-reactivity analysis. So this has been made possible with our development of the Human Epitome Microarray. This covers almost 30,000 human epitopes of the immune epitope database. And these epitopes are coming from the Immune Epitope Database, which is a free resource, NIH funded and developed at La Jolla Institute of Allergy and Immunology, based here in San Diego. So in our chip, we include epitopes for infectious disease, autoimmune, vaccine, cancer, and allergens. So as a case study, we took a commercially available human antibody anti-c-Myc, and we challenged it on our array. And hopefully you can see it. There's few but very clear hits that are seen in red and that are not c-Myc. So this is, I'll just show an example of the actual raw data. This is what you would get. Again, just one chip, but a lot of information. We sorted based on decreasing signal intensity. So here is a column of the peptide, signal intensity, and then it also gives the source of where these peptide hits are coming from. And for this circumstance, it's CMYC was reacting against some peptides in malaria, TB, hepatitis B virus, and measles. And then we wanted to get a little bit more in-depth view to see if there was any sort of consensus motif. And so we took the top 10 hits and employed bioinformatics to get more in-depth view. So I won't go into all the details in the interest of time, but for example here, for position 2, clearly a lysine is absolutely essential for binding of CMYC. So this could give you information to understand what possibly might be in your sample or where CMYC could actually cross-react. So again, one assay, but a lot of, an enormous amount of information. So that's it. So just in summary, we offer a comprehensive toolbox to validate your antibodies that's precise, fast, and low cost. We're very interested in working with the researchers, and we're based on science, and we have a bunch of scientists working here, and we all find this is really important. That's why we did the webinar, to kind of take part in telling people about kind of the issue out there with commercially available antibodies, but also that we have a solution that's not going to take too much time or money. If you have any more questions, depending on where you are in the world, we have different sales representatives, so you can go to our contact page, or you can contact our team internally directly. If you just want to look around on your own, we have tutorial videos, webinars, and application notes for review. Please sign up for our newsletter so you can hear updates, and we are on social media, of course. So thank you all for joining us, and at this time I'll take some questions.
Yes, thank you, Jill. So again, to the audience, if you have any questions, please type them in the questions window. They show up here, and I can then read them out aloud to the audience. I've been already emailing some of the participants. So one very general question here that came up, Jill, maybe you can briefly comment. I'll also open up to our panelists over our PEPperPRINT headquarters, Julia Noack and Silke Weissenberger. So how many peptides can you print on one array? That was one question.
I'm going to have our internal team kind of give their spiel.
Okay. Julia, Silke, do you want to comment on this briefly?
Yeah. So hi, here's Julia from Heidelberg headquarters. So we can print on our standard arrays 4,290 peptides in duplicate. But depending on your array size, number of peptides that you can print, so this varies from project to project, we can also print multiple array copies on one chip, and these array copies can be assayed individually with appropriate incubation trays.
Okay, great. Another question, also a simple one here. How long does it take to print one array?
Well, it would take around 1 month.
Mm-hmm.
This month, so here's Silke, also from the headquarters in Heidelberg. This month includes also quality control steps, yeah, and then the chips are ready for sending, or customers can send us a sample, and we do the staining and analysis.
Okay. Then here's another question that came up from the audience. I'm reading that. Is either the, hold on, it slipped again. Is either the conformational or linear arrays able to detect discontinuous epitopes? So the question of one of the participants is whether we can also do discontinuous epitopes. Can anybody comment on that?
It's okay, I'll give that to you.
So for discontinuous epitopes, it depends then, of course, on the nature of the epitope. So they're really challenging, I have to say. So we cannot guarantee that it would work, but we would use conformational epitope mapping and probably also a combination of linear and conformational epitopes. So in some cases we already had success with that, but we cannot predict beforehand if we will be able to map discontinuous epitopes.
Mm-hmm. Okay, so here's another question from the audience. How can I check whether my antibody binds to a linear epitope?
So we recommend the testing of the antibodies in a Western blot under denaturation conditions. And if this works in these conditions, then it's likely that the antibody recognizes a linear epitope that we can map with the linear peptides.
Okay, great. So I just want to announce this to the audience. There's a lot of questions coming in today. We won't, may take one or two more questions, but rest assured that we will answer all the questions offline and send you some detailed information. You can also check the website. Here's someone from the audience asks, could you explain again the red and green ribbon as controls? What are they? Maybe anybody of the panelists who can briefly comment on this?
Yeah, so what you see there are our control peptides. So we print HA and flag control peptides around each peptide library just to control for the staining. So in green there you see HA and in red the flag, which are then stained with specific antibodies that are fluorescently labeled.
Okay, great. So I'm going to read two more questions and then we're starting to conclude this webinar. Here's another one. Have you tried other cyclization strategies beside disulfide bonds, e.g., in parentheses here, the questioner asks, for instance, thioether or amide bond. So again, I read it again. Have you tried other cyclization strategies beside disulfide bonds? And in parentheses, thioether or amide bonds.
Yeah, we use two different kinds of cyclization processes. So the disulfide process and the thioether method, where we print the cysteine as a first amino acid on the linker, which has a specific label, and this reacts with the N-terminus of the peptides to form the bond.
Okay, great. And last question.
Kirsten, I just want to mention too, we do have an application note in the website for the conformational epitope mapping that kind of clearly describes the whole process as well.
Yeah, again, a good resource is always the PEPperPRINT website. There's a lot of information posted there, so available for download. And perhaps here the last question that I'm going to ask is, what is the maximum number of amino acid residues that you could make? So meaning, I think the participant is interested how many amino acids we can bind together on the peptide array. Anybody can comment on that?
Yeah, so I mean, routinely we synthesize 15 mer peptides, but we already showed successful synthesis of 25 mer peptides. So
Great.
This is the maximum that we successfully synthesized.
Okay. Great. So I'm going to start and conclude this webinar. So again, I want to thank everyone for attending this webinar presentation. We also wanted to thank again our speaker, Dr. Jill Rowan. We will make this presentation, these presentation slides and the webinar file available shortly on the PEPperPRINT website. If you do have any questions, please contact the PEPperPRINT team directly. And by the way, again, If any questions remain unanswered, we will do so offline and send you detailed information. So please check again our website for updates, future events, webinar announcements, and also follow us on social media, particularly on LinkedIn. So I will end this webinar now. Thanks again all for dialing in, and I look forward to see you next time. Thank you.
Thanks, Karsten.
Thanks, Jill.